everyone, I am Dr. Deepika Malik. In this video, we start with the second part of the chromatography techniques. In the first part, I gave you an introduction about the chromatography, its different types, and detailed discussion about planar chromatography, which included thin layer chromatography and paper chromatography. Now coming to the second type of chromatography based on the methods of holding the stationary phase is column chromatography. In column chromatography, the stationary phase is packed into a glass or metal column. It is used to determine the number of components of a mixture and used to separate and purify substantial quantities. It is comparatively time consuming and tedious, especially for large samples. It can be categorized as ion exchange chromatography, gel filtration chromatography, affinity chromatography, high performance liquid chromatography, and gas chromatography. Now before discussing these types one by one, there is a need to understand some important terminology related to column chromatography. Sample is the solution which is injected into the column for separation. It contains the analytes or compounds of interest. Analyte or solute is the individually separated components from the sample mixture. As you can see in the diagram on the right side, the sample mixture in a beaker which contains different analytes or solutes which need to be separated. Stationary phase or adsorbent is a porous solid matrix through which the sample contained in the mobile phase percolates. It may be a solid or gel that is immobilized or fixed inside the column. Mobile phase or carrier refers to the mixture of the substances to be separated, that is analytes, dissolved in an eluent or solvent. It is passed over or through the stationary phase after the sample mixture of analytes which are to be separated has been applied to the column. The sample mixture is basically poured over the stationary phase after it is settled into the column, after which mobile phase is applied. Eluent is a solvent used in order to effect separation by elution. It moves the analytes through the column. In liquid chromatography, the eluent is the liquid solvent. In gas chromatography, it is the carrier gas. So as you can see, the form in which the eluent is used forms the basis of liquid and gas chromatography. Elution is the process of washing out a compound through a column using a suitable solvent or eluent. Eluate is the fluid or mobile phase that is exiting the column that is collected in the test tube as shown in the diagram. Eluate is the analyte or solute of interest exiting the column. So to summarize, let us see the table. Sample is basically solution which contains the analytes which are to be separated. Analyte is the solute which is present in the sample solution entering the column. Stationary phase is a porous solid immobilized matrix which is packed into the column. Mobile phase is basically the eluent or solvent containing the analytes. Eluent is the solvent which may be a liquid or gas entering the column. Eluate is the solvent leaving the column and eluite is the solute leaving the column. The diagram gives an overview of the working of a column chromatography. Here the sample mixture which contains three types of analytes is applied onto the column. First, the stationary phase is packed within the column, and then the sample mixture is applied over which the eluent is poured. This eluent picks up the analytes from the sample mixture and starts percolating through the stationary phase and passes out of the column. The analytes get separated into different bands, as you can see, red, purple and yellow bands, depending on various parameters which forms the basis of different column chromatography. Now let us see basic components of column chromatography. A typical column chromatographic system using a liquid or gas mobile phase consists of the following components. First is the column. In liquid chromatography, these are generally 25 to 50 cm long and 4 mm internal diameter and made of stainless steel, whereas in gas chromatography they are 13 meter long and have 24 mm internal diameter and are made of either glass or stainless steel. They may be either of the conventional type filled with a stationary phase or of the microbore type in which the stationary phase is coated directly on the inside wall of the column. There is a stationary phase which is chosen to be appropriate for the analytes to be separated. A mobile phase and delivery system chosen to complement the stationary phase and hence to discriminate between the sample analytes and to deliver a constant rate of flow into the column. an injector system which is used to deliver test samples to the top of the column in a reproducible manner, a detector and chart recorder to give a continuous record of the presence of the analytes in the eluate as it emerges from the column. Detection is usually based on the measurement of a physical parameter such as visible or ultraviolet absorption or fluorescence. A peak on the chart recorder represents each separated analyte. So basically the detector detects the analytes and represents the data in the form of chromatogram. A fraction collector for collecting the separated analytes for further biochemical studies. Now let us see the steps involved in column chromatography. First is the packing of the slurry on column. Second is sample loading. Third is elution. Fourth is detection of components. And fifth is isolation of compounds. These steps are common for all types of column chromatography. Now we will be discussing all these steps one by one. First is the packing of the slurry on column. Slurry is basically the stationary phase powder suspended in the mobile phase. It is then stirred well to drive off all the air bubbles. The resulted semi-solid slurry is then poured into the column. A cotton gauge or a nylon gauge or a wool gauge is set at the bottom of the outlet so that the stationary phase do not leak out of the column, as you can see in the image. After slurry application, the column is allowed to settle overnight. In this way, stationary phase is immobilized into the glass or metal column. The physical nature of stationary phase puts resistance to the flow of mobile phase as it moves down under gravity, which is known as back pressure. The table summarizes the effect of size of stationary phase particles. If the size of stationary phase particles is large, then the surface area to volume ratio will be small. Back pressure generation will be low, so the flow rate will be fast and therefore the resolution and performance will be low. Whereas if the size of the stationary phase particles is small, the surface area to volume ratio will be large, back pressure generation will be high and therefore flow rate will be slow, and this will give a high resolution and performance. So more the resistance by back pressure, more will be the resolution. Now after the settling of the slurry into the column, next step is the sample loading. Two different methods are used to load sample into the column, that is wet method and dry method. In wet method, the sample containing the analytes is applied over the stationary phase. The sample is dissolved in the minimum amount of solvent, usually 5 to 10 drops. Using a pipette or syringe with a thick needle, the sample is dripped directly onto the top of the silica. Do not disturb the surface. Ideally, the sample should be equally distributed across the surface so that it forms a thin horizontal band. Once the entire sample has been added, it is absorbed within the column, and now we can begin with the elution process. In dry method, the sample to be analyzed is dissolved in the minimum amount of solvent and about 100 mg of silica gel is added. The mixture is swirled until the solvent evaporates and only a dry powder remains, which is then transferred on the top of the prepared column. Once the sample is in the column, fresh eluting solvent is added to the top and we are ready to begin the elution process. Sometimes a layer of sand, approximately 2 to 5 mm, is added on the top of the stationary phase. This helps to prevent the surface of the silica from being disturbed as sample or solvent is added into the column. You can see the illustration of both the methods in the images shown. After sample loading, next step is elution. Apply the eluent over the sample so that it starts percolating the analytes through the stationary phase and finally out of the column. Eluent consists of an organic solvent, mix of solvents, or a buffered aqueous system. Different analytes get separated as individual bands within the stationary phase based on their partitioning between mobile phase and stationary phase. Substances exhibit different mobilities by reason of differences in vapor pressure in case of gas chromatography, in molecular size in case of gel filtration chromatography, or ionic charge density in case of ion exchange chromatography. These different properties form the basis for different types of chromatographic techniques. Elution can be of two types: isocratic elution or gradient elution. In isocratic elution, the components of mobile phase is constant, as in gel filtration chromatography. That is, the mobile phase which is used for elution is same throughout the process. In gradient elution, the components of mobile phases changes gradually. For example, change in pH or salt concentration of buffer is done in case of affinity and ion exchange chromatography, which means that a solvent or buffer of different pH and salt concentration are used one by one for eluting the solutes or analytes from the column. Now once the eluate containing the separated analyte comes out of the column, it is then detected. For this, a detector is attached to the column which records time and relative quantity of analyte, that is elution volume, collected in the tubes. Fixed amount of fractions are collected in a fixed amount of time in different test tubes. For every column there is an optimal flow rate. In laboratory scale, the tubes are changed at a fixed rate manually, whereas at larger scale, the tubes are changed with an automated system. The fractions collected in tubes are analyzed using a spectrophotometer by taking their optimal density at respective wavelengths. The elution pattern is depicted in a graphical form called chromatogram, as you can see in the second image. The first image shows the effect of flow rate on the resolution of the chromatographic process. If the flow rate is too slow, diffusion process will lead to band widening, whereas in optimal flow rate, it gives a sharp band. And if the flow rate is too fast, there is not enough time for equilibration and the compound will be forced down the column, leaving a long tail behind. Now the last step is the isolation of compound. The fraction of solvent containing the desired compound is isolated by evaporating through rotary evaporator. In this apparatus, as the solvent boils, the evaporated solvent filters up through a cold trap and condenses into a collecting reservoir. The sample remains at the bottom of the flask during evaporation. In this way, we get the final product, that is our solute, separated from the sample mixture. Now let us see what is sample derivatisation. Derivatisation is the process of chemically modifying a compound to produce a new compound which has properties that are suitable for analysis using chromatographic technique. The chemical structure of the compound remains the same and just modifies the specific functional groups. Some functional groups, especially hydroxyl, present in a test analyte may compromise the quality of its behavior in a chromatographic system. The technique of analyte pre or post column derivatization may facilitate better chromatographic separation and detection by masking these functional groups. Derivatization is able to introduce new groups into sample molecules in order to increase their sensitivity to UV absorption and fluorescence detection. The table shows few examples of analytes which are modified by derivatization by using the corresponding reagents. For example, the separation of alcohol, amines and phenols is made better by using the reagent 3,5-dinitrobenzoyl chloride. For better separation of amino acids and peptides, we use phenyl isothiocyanate and tensyl chloride. For carbohydrates, we use benzoyl chloride. For carboxylic acids, we can use 1p-nitrobenzyl N-NO diisopropyl isourea. For fatty acids and phospholipids, we can use phenanyl bromide or naphthyl bromide. Now we will be discussing the different chromatographic performance parameters. All these factors are very important from the performance point of view for chromatographic techniques. These parameters include distribution or partition coefficient, chromatogram, retention time, dead or void time, void volume, adjusted retention time, peak characteristics like peak width, peak area, peak height, asymmetric peaks including fronting peak or peak tailing, relative retention time, retention or capacity factor, selectivity or separation factor, and resolution. Now we will be discussing all these parameters one by one. First is the distribution or partition coefficient, also designated as KD. Chromatography is based on the principle of distribution or partition of the solute, that is analyte, between two immiscible phases, that is stationary and mobile phase. The relative interaction of solute with these two phases is described as distribution or partition coefficient. Individual analytes have different Kd values, and because of this difference in Kd, analytes move along the mobile phase at different rates. For two such phases, that is stationary and mobile phase, the value for the distribution or partition coefficient is a constant at a given temperature and is given by the expression concentration of analyte in stationary phase divided by the concentration of analyte in mobile phase. The phases are chosen such that the components of sample have different solubility in each phase. If one component is sufficiently soluble in stationary phase, it will take longer time to travel through a column than the other which is not highly soluble in stationary phase but more soluble in mobile phase. Due to this difference in mobility, one component will be separated while it travels through stationary phase. Now, to understand further parameters, we need to revise about the chromatogram. A chromatogram provides gross information about the number of analytes present in the sample mixture and time at which individual analyte emerge out of column during the process of separation. For optimal separation, there is visualization of appearance of sharp, symmetrical chromatographic peaks in a chromatograph. A chromatogram is a pictorial record of the detector response as a function of elution volume or retention time. It consists of a series of peaks or bands, ideally symmetrical in shape, representing the elution of individual analytes as shown in the figure. In the graph, we can see the elution of component B at time t1, component A at time t2, and component C at time t3. This means when the sample was applied onto the column, the sample mixture got separated into its different analytes, and first component B came out of the column, then component A, and then component C. Difference in height of the peak indicates the different quantity of analytes present. Now we have understood what a chromatogram is. Let us understand other parameters. Next is the retention time. It is the actual time between the sample injection and detection of an analyte peak reaching detector under set conditions as indicated in the graph. It is always more than the void time. Now what is dead or void time? In addition to the peak for the solute, the chromatogram shown also includes a small peak that elutes shortly after we inject the sample into the mobile phase. This peak is for non-retained solutes. The unretained solutes do not interact with the stationary phase. They pass the column through the free spaces or void volume between the particles of the stationary phase at the same rate as the mobile phase. The time required to elute non-retained solutes is called column's dead or void time. No peak can be seen before the void time. The value of dead or void time will be same for all analytes as the void volume is same in a column and can be measured by using an analyte that does not interact with the stationary phase but simply spends all of the elution time in the mobile phase traveling through the void volume. So to sum up till here, retention time is the time for analytes which are retained by the stationary phase, whereas dead or void time is the time taken by the non-retained solutes as well as the solvent which do not interact with stationary phase and move out of the column. So the dead or void volume is determined by the first liquid that is coming out of the column. Next is the void volume. The volume of the free space is referred to as column void volume. Adjusted retention time. Actual retention time is the time taken for a component to travel through the column. However, this cannot be less than the dead time as I told earlier also. This is obviously because retained analyte will take more time than compared to the non-retained analyte. Therefore, the adjusted retention time is calculated, which is the time the stationary phase retains the analyte. This time is characteristic of the analyte and is the difference between the observed or actual retention time and the dead time. So when we subtract the dead or void time from the actual retention time, it will give the time when the analyte is in actual contact with the stationary phase. Next slide shows the peak characteristics. First is peak width. Peak width gives the efficiency of the column when combined with retention time. If the flow rate is too slow, which means that the longer a component stays on the column and more is the retention time, the wider is the peak. The band is dispersed as it travels through the column. Dispersion leads to band broadening and wide peaks. Peak width is measured by extending tangent lines from both sides of the chromatographic peak, as you can see in the figure. Next is the peak area. Peak areas are used for most quantitative chromatographic estimations. Peak heights can vary due to distortion of the shapes such as broadening or fronting and tailing. However, in such situations, areas are not affected and show high reproducibility. Another parameter is the peak height. The height of each peak is in proportion to the amount of the particular component present in the sample mixture injected into the chromatograph, but as I told earlier, the peak height can vary and therefore peak area is a better option for quantitative measurement in chromatography technique. Next slide shows the asymmetric peaks. Asymmetrical peaks often result due to fronting peak or tailing peak. This leads to error in reporting of areas, resulting in lowering of precision and accuracy of quantitative measurements. Tailing peak, which occurs when some sites on the stationary phase retains the solute more strongly than other sites. As a result, the solute comes out of the column for a much longer duration and causes tailing of the peak. Whereas fronting peak is the result of overloading the column with the sample. Due to overloading, all the analytes do not get bound to the stationary phase and starts eluting out of the column before their time. Next parameter is the relative retention time or RRT. It is common practice to relate the retention time or adjusted retention time for an analyte to a reference internal standard. In such cases, the relative retention time is often calculated as retention time for the analyte divided by the retention time for the standard. Retention factor or capacity factor. The migration rate of an analyte through a column is measured by this factor. It is simply the additional time that the analyte takes to elute from the column relative to an unretained or excluded analyte that does not interact with the stationary phase. The retention factor for the unretained analyte is 0. The following are the expressions for the calculation of the retention factor. It can be calculated by retention time minus dead time divided by the dead time, or adjusted retention time divided by the dead time, or mass of analyte in the stationary phase divided by the mass of analyte in the mobile phase, or distribution coefficient multiplied by beta, which is equal to volume of stationary phase divided by volume of mobile phase. Now, if the retention factor is less than 1, it means that elution becomes very fast, that accurate detection of retention time is very difficult. And if the retention factor is more than 20, it means that elution takes a very long time but favors a good separation. Ideally, the retention factor for an analyte should be between 1 to 5. Another parameter is selectivity or separation factor, denoted by alpha. It can be defined as the relative ratio of two analytes which describes their separation. Therefore, if a sample mixture contains two analytes A and B, the selectivity or separation factor can be calculated as retention factor for analyte A divided by retention factor for analyte B. Or it can be calculated by distribution coefficient for analyte A divided by distribution coefficient for analyte B, or by adjusted retention time for analyte A divided by the adjusted retention time for analyte B. Now, for separation to occur, alpha must be greater than 1. If alpha is maximized, good separation is achieved, but analysis time becomes too long. To improve separation, alpha can be manipulated or increased by following factors, that is, by change in mobile phase composition, or change in column temperature, or by change in composition of stationary phase. Now let us discuss resolution. The success of a chromatographic separation is judged by the ability of the chromatographic system to resolve one analyte peak from another. Resolution is the degree of separation between peaks. Resolution is defined as the ratio of the differences in retention time between the two peaks to the mean of their base widths. TRA describes the retention time for analyte A, TRB describes the retention time for analyte B, WA is the base width for analyte A, WB is the base width for analyte B, and WM is the mean of the base widths for analyte A and B. Now when resolution is equal to 1, separation of two peaks is 9.7% complete. And when the value of resolution is equal to 1.5, it means overlap is reduced to 0.2%. Now as you can see in the chromatogram shown, the blue peaks are very sharp, which means that there is high efficiency of separation by the chromatographic system, whereas the peaks in red shows unseparated peaks or non-sharp peaks, which shows low efficiency. Now many factors influence the final resolution, that includes the sample volume applied on the column, ratio of sample volume to the column dimensions, particle size of stationary phase, particle size distribution of the analyte between the two phases, packing density, pore size of the particles, flow rate and viscosity of the sample and buffer used. So till now we have discussed and established our base for the understanding of column chromatography working.